All right, welcome everyone again for another seminar. Thanks everybody for turning up again with such a big crowd. It's my pleasure to introduce Michael Butts today for this seminar. We've known each other, well, I've known you at least since I started in this evolution and ecology of infectious diseases field. And then we really started interacting a little bit more when he published this marvelous paper a couple of years ago on using evolution more or less to our advantage. So since all these pathogens and insects that we try to control are evolving anyway, why not just use that evolution to our advantage and make them evolve to something that's going to be useful to us? And he's going to talk to us about that. So just a little bit of background on Mike. So he did his PhD at the University of Liverpool on disease ecology, and then he went for quite a while on some fellowships to Japan. And in 2000, he got a NERC fellowship to go to the University of Sheffield, where he eventually stayed until where he became a full professor. And then in 2011, he moved to Exeter, University of Exeter. And in 2015, he fled the UK just in time and went to UC Berkeley, where he is ever now. So. Yeah. Floor is yours.
Thanks, Sylvie. It's great to be here. Yeah, I didn't predict Brexit by leaving, but I would have thought it would have been resolved by now. But no, it keeps going on. So today I'm going to give you a fairly broad view that kind of illustrates the sort of different approaches that we take in the lab. And we work on infectious disease, and we work on ecology and evolution, and we use theory. We have model systems, and we apply the theory. And I want to give you a sense of all of that. And I'll finish by talking about this work of where we might be able to use evolution to our advantage in terms of controlling vectors. I want to start with acknowledgements. I've started always putting acknowledgements up early in the talk because all of this work is really collaborative. And it involves a bunch of mathematicians like Ben and Andy and Roger. So I trained as an ecologist and an entomologist, but I got really interested in ecological theory and then had no idea how to do maths. And so it's very challenging at that point. You're sort of kept outside of being able to contribute in that field, partly by just not having the language of mathematics. And so what my approach to it, and I guess approaches could be to go and take courses and things like that. In the UK, we don't have any courses when we do a PhD. You come in 3 years, do your PhD. So my approach was actually to go and bother a mathematician, which was Roger Bowers, who's extremely patient and helpful. And that's how I ended up and doing so much theoretical work, just by the help of Roger. And then the work I'm going to talk about on vectors is really driven by Penny Lynch, who's an amazing person who's moved from the world of finance into the world of mathematical biology. And then we also do a whole bunch of empirical systems with people like Angus Buckley and Ed Zawestra. Lewis is a bee guy, and Elisa is in my lab and works on the moth system I'm going to talk about. So this is a lot of people's work. So we work on infectious diseases. Why do we work on infectious diseases? Almost why wouldn't you work on infectious diseases? I mean, there's huge impacts. We know about huge impacts in the past. I always put this in my introductory infectious disease lectures. And this is an estimate of the world population as we head towards the Black Death. And you can see these huge significant impacts on human population in the past of infectious disease. We also know that there's still major issues with infectious disease. We think about endemic diseases like malaria, but 2014 made us realize that you could have very, very large outbreaks for which the world's perhaps not at that time at all prepared. And the 2014 outbreak of Ebola in West Africa really showed us that we weren't prepared and we weren't able to deal with infectious disease in a new context. And there's also this whole question of potentially future infectious diseases. Where are they going to come from? Are they going to be hypervirulent? Things like SARS seem to come from nowhere. And we have, this is Kara Book is in my lab, is a Miller Fellow in my lab, and she's working on viruses in bats. Bats are a unique source of highly virulent zoonotic viruses, and we're trying to understand why that is. And we're actually also trying to ask that very, very hard question of, can we do anything to try and understand the prediction of an emergence of new infectious diseases? It might just be random, but it's such a big problem that it's important that we can try and develop some evolutionary ecological theory and approaches to try and understand that problem. It's quite possible that one of these diseases is just going to pop out, jump into the human population, and then just spread rapidly around the world. And it might just be bad luck that it's highly virulent and highly transmissible. So understanding those problems is still a major reason to work on infectious disease. So we work on infectious disease because they matter to humans. There's potential for them to matter in the future to humans, and we're not really very well prepared. I don't think we have very much evolutionary theory in particular on what we would expect for emergent disease. Disease is also hugely important in agriculture. Any of you in the UK, when there was the foot and mouth outbreak of disease in 2001, Sylvie was there in Edinburgh, I guess, you would see these pyres, funeral pyres, where they were just burning this huge number of cattle in order to deal with this foot and mouth outbreak. The estimated cost UK was £8 billion, and when you think that the UK science budget was the order of £1.4 billion, you can understand why infectious disease and understanding infectious disease is really important. And actually, I'd argue that the modelers who came in during this outbreak had a really big impact on controlling this disease. Just 5 or 6 postdocs was very, very useful in this case. And then, of course, the other thing is just ecology, you know. So this is the example I also use in my undergraduate lectures to try and get people interested in infectious disease. Think of the Serengeti in Tanzania. So this is the ultimate predator-prey system. This is lions and wildebeest. Well, not really. So they did widespread rinderpest vaccination of cattle on the edge of the park. So they're not vaccinating any of the wildebeest or anything in the park, just the cattle that are on the edge of the park. And although no wildebeest were vaccinated, their numbers increased 8 times. So actually it was the parasite and the spillover parasite from the cattle that was actually the thing that was maintaining the population size of these prey, not the predators at all. And these guys took off. We actually ended up with loads more lions, and then the lions got canine distemper virus because they were at higher density and went down. So actually, disease really matters in natural systems. It's just often not as obvious. Let me give you an example of that from our own work. So we do some work on deformed wing virus in honeybees. And this is a paper we published led by Lena Wilfer recently, which was basically just an analysis using phylogenetic methods to just try and work out where this disease had come from. And it was very clear that this is a disease that is spreading around the world due to the fact that we're taking our bees all around the world. So this is a disease that's spreading, and in a new emergent disease in bees due to apiculture, and it has this global spread. So this is an example, and I think it's an example where agricultural practice in this case leads to worldwide spread of disease extremely quickly. And this, I think, is typical of what's happening with ecology of infectious disease now, including human diseases as they emerge. So one of the things that's very interesting about deformed wing virus is that it's associated with varroa mites. So varroa mites crossed over onto Apis mellifera, the western honeybee. And then once they started infecting the western honeybee, you suddenly got this huge increase in lots of viruses, but in particular deformed wing virus. So deformed wing virus gained a new transmission mode. It started being vectored by this mite. And then if you've read Paul Ewald's books, and many of you have, there's a prediction that once you start becoming vector-borne, you'll be selected to become more virulent. Okay. And that's exactly what we've seen in deformed wing virus. So this is an example of an emergent infectious disease that has occurred due to gaining a new transmission route, and then it's evolved to have much higher replication rates within bees, much higher virulence, and it now causes disease. It causes deformed wings. Okay. So this is an example of emergence. Emergence by gaining a new transmission mode in a particular focal species. But we were also interested to see whether there's actually then knock-on effects of this emergence into other species. So this is what we did in this, effectively what a natural experiment that we had in the UK. This is the UK, this is France, this is Europe, this is soon not to be Europe. And what we have is a whole series of islands off the coast, and then we have mainland sites and we have different islands. And some of those islands are still free of the varroa mite. And then some of those islands have the varroa mite, and then the varroa mite is on the mainland everywhere you see. So we effectively have a natural experiment here. We can look on the islands where we don't have varroa mite, and we can look to see what's happening with deformed wing virus, and we can compare that to areas where the varroa mite has invaded. And what we wanted to look at was, one, okay, what happens when you have varroa mite to the virus in the honeybee? This is something we've been looked at in other places, and it tends to, as I said, increase the replication rate and the virulence of the virus in the honeybee that has these varroa mites. But we also wanted to see if there were any knock-on effects to the virus in the wider pollinator community. So the important thing to note here is that varroa mites are not infecting, are not on any of these bumblebees, on these natural pollinators. They're only on the honeybee. But by them being on the honeybee, an increase in virus in the honeybee, is there any impact in the wider community? So this is work that was carried out by Robin Manley during a PhD with Lena and I. Okay. And what we see is that there's a very clear effect. First of all, if you look, this is the honeybee, this is mellifera, and if you look at the prevalence of deformed wing virus in islands without the varroa, so these are varroa minus islands, it's much lower than when there is varroa, either on islands with the varroa or on the mainland, which always has the varroa. So again, it's another example. Not only do you get higher viremias within individual bees, there's much higher prevalence of deformed wing virus in bees when you have the varroa mite. Okay. So the deformed wing virus is there, but low prevalence without varroa mite, much higher prevalence with the varroa mite. But what's really interesting is you're getting this same effect, not as strongly, but you're getting this same impact in these bumblebees as well. That there's higher prevalence of deformed wing virus into the bumblebees when there's varroa mites on the honeybees. Is this just about connectivity? One of the nice things about this system is you could argue, okay, well, there's just something weird about those islands, right? Certain islands that don't have varroa mites on them, maybe they're the islands where varroa mites can't get to because no one ever goes to those islands, and that's why there's less disease generally. Right? But we've got a nice control for that because these bees have lots of diseases. So we can look. We also looked at nosema. Nosema does no interaction with the varroa mite at all. Okay, so you wouldn't expect any interaction with the varroa mite. And you don't see any particular difference in nosema between islands with and without the varroa mite. So that's saying there's not something special about the islands without varroa mite apart from the fact that they don't have varroa. Okay. So I think what this study highlights is that you can get some very complex knock-on effects from infectious disease emergence in a focal species, whether that's in this case, the honeybees in some sense is an agricultural species. What's happened is actually an evolutionary emergence in that there's been a new route of transmission that's come about due to this vector moving on to the honeybees. That's then led to much more rapid replication of the deformed wing virus, and deformed wing virus comes at much higher prevalence due to the vector in that species, and then this knocks on out into the wider community. And I think honeybees and their viruses are very important just in their own rights. Many of you heard about colony collapse disorder, and we're worried about natural pollinators, but they're also a fantastic model system, the sorts of things that we might expect when we get the emergence of new infectious diseases. You get an emergence of infectious disease in agricultural population. It's then likely to spill over and have knock-on effects into natural populations. So I think we really need to understand these basic drivers of emergence in infectious disease. So that's kind of ecological impacts and community-level knock-on impacts of disease emergence. The other thing we're really interested in is evolution in infectious disease. So a few years ago, it was a Darwin year, and I went to lots and lots of talks, including many at Edinburgh when Sylvie was there, where people were talking about Darwin. And it's very noticeable to everybody that Darwin had basically thought of everything. He really was very prescient about many, many fields in evolutionary biology. But Yanis Antonovitz put together these couple of slides that made this really interesting point, which is that Darwin wasn't interested in infectious disease at all. Right? There's really very little, if any, mention of it in the origin of species. And this is kind of, well, it's very interesting and slightly odd as well, because he's Victorian man, so he's seeing the effects of infectious disease in terms of mortality all around him all the time. And this is this letter that Yanis found, where it's a letter from Darwin to Hooker, Kew Gardens, and actually he was just talking about botany and what was happening here, and he's having a nice talk about that. Then he just mentions one of his daughters was, you know, extremely ill. It was a form of typhus fever, but she's okay now. And then he's straight back into, has this particular plant ever set seed at Kew? So there's no link really to the fact that actually infectious disease could be really important in terms of evolutionary outcomes, despite the fact that you're seeing it in humans all the time. And I think a lot of this is because people were not looking, it wasn't easy to understand and see infectious disease in nature. So really, infectious disease is ignored by evolutionary biologists pretty well until Haldane's paper in 1949, which is absolutely fantastic paper, if you can read this paper. It talks about coevolution of hosts and parasites, the role of parasites in rapid speciation, speculating the role of genetics in human disease resistance. And lots of these things are just coming back into fashion now. But really, it was only that paper where anyone started talking about evolution and infectious disease. I think it's also true to say it's pretty well ignored by ecologists until May and Anderson in 1978. Everyone was building predator-prey models. They weren't building host-parasite models until the late 70s and the 80s. And Anderson and May were very influential in building disease ecology. Okay. So I'm going to give you some examples about why, well, I hope I convince you that ecology and evolution of infectious disease is important. I think in natural systems it's often overlooked because it's difficult to see. But we all understand the importance of infectious disease to humans and in agricultural systems. So what does my lab do? So we're interested in infectious disease, ecology, and evolution. And we build a lot of theory. We build theory which is basically ecological, and if it's ecological theory, equations look a bit like that. And we build theory which is very evolutionary. If you're a population geneticist, the equations look a bit like that. But what we're really interested mostly in the lab is this interface, actually, is understanding the way in which ecology feeds back into evolution and evolution feeds back into ecology. So I'm going to talk about a bunch of theory on that and how actually these eco-evolutionary feedbacks are really important to determine optimal outcomes and also important to determine diversity. So that's the sort of theory we do. We use model systems, and I'm going to talk to you about the way in which we use this model system to test some of the assumptions of the theory. We use this as an insect virus system. Then we also use bacteriophage systems typically to test the predictions of these theory. Very good to do experimental evolution in these systems. And then we also look at model systems in the field as well. Best model systems in the field are typically plants, because you go out, you can look and see if they're diseased, and you go back, you can find them again because they haven't gone anywhere. So we also work on some plant systems. And then finally, so I want to give you one example of this. We try and apply these theory to specific systems. So I said that the honeybee virus system is a great model system. It's also extremely important system. And when you think about, so in California, and you think about how many millions of bees are brought every year to California for things like the almond crop, they're brought from all over the US, everything's churned together. What is happening with all the diseases in that system and how important they are? And then we apply models to different specific diseases. We've been working on TB in badgers in the UK recently. Okay. So let me start by talking about how we use model systems to test the assumptions of the theory. So one of the things I'm very interested in is trying to understand optimal defense and the evolution of resistance, the evolution of tolerance to parasites, and optimal immunity. And when you look at most evolutionary models, they assume fundamentally that things like resistance are costly in some sense, so that you can't be completely resistant to a parasite without there being some other cost in terms of your life history. But actually, showing those costs in many systems is difficult. And one great system in which we can look at things like cost to resistance tend to be insect pathogen systems. Okay. So this is one of our models. This is the Indian meal moth, Plodia interpunctella. This is a healthy larvae, and this is a larvae that's infected with a granulosus virus. So one of the reasons we worked on this system was because it's extremely easy to tell when something's infected. So they turn white. It's also extremely easy to show what the fitness effects and costs are of being infected. They turn white and blow up and die. So they clearly, it's bad for them. Okay. Actually, I say that's a healthy larvae. I think it's pointed out to me recently it's dead, but it's not infected. Okay. So it's very clear to say when they're infected. They live within, they're a stored product pest, so they live within basically anything. They're close to spontaneous life. I found them in tea bags at some point. They could move through, eat your tea bags. They're very, very easy to keep in large numbers. We can keep them in these microcosms. We can keep them in these cake-bot microcosms. And one of the things we can do with this system is we can run the host and the parasite together over many years, basically indefinitely. This is our high-throughput technology. We bought KitchenAid mixers for the food. It kind of completely doubled the amount of insects we could deal with. So it's a super, super simple system. We just mix up some baby food and different sorts of oats, whatever you like. We can control the system very, very tightly in terms of its resources. And critically, we can run long-term selection experiments, long-term population-level experiments, where we have the host and the parasite together. And that's really hard to do. There's actually relatively few experimental systems, because what tends to happen is you bring in a pathogen and it just infects everything. You can't keep anything clean. Or you bring in a pathogen and they just drop out all the time and you lose them. There's very few systems where in the lab you can manage it and you can maintain it over many years. So we can do that. So we've been working on this question of cost to resistance in this system for a while, since my PhD. So what we did was set up a selection experiment. And what we did was we set up a whole bunch of populations. And in those populations, we either mixed the virus into the food or we didn't. So we were selecting for resistance in those populations where we added the virus to the food. These are the populations. We can do lots and lots of them. And actually just with one technician or 1.5 technicians, you're able to maintain a very large number of replicates. So we stick in 50 individuals, they run through, a month later, we take them off, we put them back in, and the food either has virus in it or doesn't have virus in it. We can select for resistance, and then at the end of 10 months, the generation time is about a month, so after 10 generations, we can examine to see whether resistance can evolve, and we can look for correlations to see if there's any cost. What we did that was a little bit different here is we did this across 8 different resource levels. So we're able to very tightly control the amount of resources that we put into the food. So we can go from very high quality food across a whole load of intermediate levels of food to relatively poor quality food. And then within those populations, we're then selecting for the evolution of resistance. So a very simple argument would be that if resistance is costly and difficult to evolve, it's going to be much harder to evolve it when you have less resources. And actually, you know, when I did my PhD, it was all based on looking for trade-offs and trade-off for resistance. And if you look at that literature, you find that often people can't find any cost to things in their labs. And often I think the reason for that is that you're keeping the animals under really, really good conditions. And by keeping them in very good conditions, you may not pick up the cost. So what we wanted to do, look at the selection experiment, see if we could evolve resistance, look for correlated trade-offs, which might be evidence for costs, but actually just ask this basic question: is it easier to evolve resistance when you have more resources? Okay. So we could do this in this system. We have multiple replicates. And so this is an assay of how resistant they are at the end of this selection after 10 months. And here I'm showing you where we're now assaying them all on a common garden on the same resource level. We also assayed them on their own resource level and on different resource levels, but the story is basically the same. Okay. And what we find, so you find the open circles here are the control populations. So they're the populations where you've been evolved on different resource levels, but there's no virus. And the dotted line shows the relationship with the percentage that are infected, so therefore how resistant you are at the end of the population, at the end of the experiment, and your resource level. One of the first things to show is that the controls, there was no effect on resistance. So if you're just in a poor resource environment, but there's no virus around, there's no evolution directly of the resource environment on your resistance. Okay. If you look at the selected lines, the ones with the virus, you can see, well, it's below this, so they're less likely to get infected. So they did evolve resistance. And you'll also notice that they evolved more resistance at the high resource levels. Okay. So we evolve resistance, and it's easier. You can evolve more resistance when you have more resources. So in some sense, that's a direct test of is it costly to evolve resistance. What's super interesting is you can then also look at correlated changes. Okay. So if you look at the development time of those individuals, so you take adults out, this is after an F2, so you let some time to avoid maternal effects, and then you grow up a bunch of larvae all on the same quality resource. Okay. What you find is, again, these are the controls. There's no selection on how long your development time is just by being in different resource environments. Okay. But you always take longer to develop if you're in the selected lines. So the resistance ones have a cost for their resistance in that they have a slower development time. Okay. The thing that's really interesting about this, for me, is that that difference, so that cost, is much higher in the ones that were in poorer resources, even though they evolved much less resistance. Okay. So they're under poor resources, it's costly in development time. They try and evolve resistance, so they pay an even bigger cost to get that less resistance. Okay. This is actually an example of where somebody repeated an evolutionary experiment. Because I did this same experiment in my PhD, but I did this same experiment in my PhD, but just at high quality resources. Okay. So just here. And again, in that paper, so it's in '93, it's a while ago now, I showed that there was resistance and that there was an increase in development time. And this is actually, I think, my most cited paper, which is great. But I was really lucky by doing it in these high-quality resources. It gave me a really good chance of showing resistance, but a really low chance of showing this cost, potentially. And I think this is a good example of where, if you're looking at trade-offs and costs, don't do your experiments under high-quality resources, because you can get the benefit, you can select much more easily on the trait, but it's much harder to pick up the costs. So, so this, I guess, is in some ways, in some ways this is one of the, I'm told you should never say that your work is really, really good, because at least this is, so we've at least shown this same cost twice in two separate experiments. And we've actually also shown it in another way as well, where we can also do inbred lines in this system. So we've got these inbred lines where we, in principle, you're taking a male and a female, and you keep breeding for male and female. So you should begin almost close to a sort of clonal-ish genotype in each line. And if you plot those different lines and those different genotypes, you again get this negative relationship between resistance and development time. So if you're more resistant, the more resistant genotypes have a longer development time. Okay. So we can show costs, and we can show costs in multiple ways, and we can show that those costs are really dependent on resources. Okay. And to me, what that suggests to is that you're gaining resistance in different ways depending on the context, depending on your resources. And this system, I think, works very well because most of the resistance in this system is due to, is probably acting at the gut, and is a lot to do with stopping the virus getting across the gut. And of course, the gut is critical for you taking in resources. So there's a very strong link between the resources and the defense in this system. And that's why you maybe see these relationships so clearly. But my sense is that these sorts of relationships are going to hold for most patterns in terms of immunity. They're going to be really dependent on context. They're going to be really dependent on resources. Okay. So just to review all of that, so we've got costs of resistance, and multiple approaches have demonstrated the same trade-off. It's very critical that these costs are resource dependent, and then potentially you're finding is that you actually can gain resistance in different ways and in different pathways depending on the resources that you have. The next thing we want to do is actually measure the shape of this trade-off. If you want to annoy any evolutionary theorist who's assuming a trade-off in their model after they've given a nice talk, ask them, what shape of the trade-off did you assume? Did you assume that costs tend to accelerate? Are there just a linear relationship? Do costs tend to decelerate? So as you get more and more resistance, it gets decreasingly costly, acceleratingly costly. All our theory assumes something like that, and that assumption critically determines the outcome, not just qualitatively but quantitatively. But we have almost no measures at all of any shapes of trade-offs in any systems. We have some systems like this. I think it's one of the best ones. It shows as a trade-off. But how does that vary? What's that relationship? And that's really important to the outcome. And we're trying to do that by having more and more inbred lines, which in principle might give us a sense of the shape of the trade-off. We've also we've got a genome, and we've looked at, I'm not going to go into this very much, we spent a lot of money on the genetics and got very little insight per dollar. So all the resistance happens at the gut, and the gut's really important. It's lots of genes. Okay. All right. So that's how we use systems to test the assumptions of theory. One of the key assumptions of theory is that defense is going to be costly. Let me tell you about a little bit of a theoretical model that we worked on, something we've been interested in for a while. So if you look at innate immunity, typically you can divide lots of patterns up quite nicely into ones that are either constitutive or induced. Okay. And this is a sort of general pattern right across different systems. If you look at plants, they tend to have constitutive and induced defenses, innate defenses. Invertebrates, constitutive and induced defenses. So constitutive defenses that are there all of the time, induced defenses that turn on after you've got infected. Okay. The same with vertebrates as well. We're very interested in this to try and understand what are the drivers of relative investment in constitutive and induced defenses, since this seems to be pretty ubiquitous. So what we can do is build a model of this. Okay. So we have a constitutive defense where you have susceptible individuals, they're going on to become infected and infectious. And what the constitutive defense does is stop you becoming infectious. So you might get infected a bit, but you've got rid of the defense before you're infected onwards. And this is what we're going to assume. Individuals die at an increased rate due to being infected. And individuals can recover. Now what we can assume for the induced defenses, the way in which we can model this, is that you're susceptible, you become infected and infectious, and having better induced defenses means that you recover at a faster rate. So you do become infected, you do become infectious, but you're investing in recovering quicker. Okay. Now for me, this is a really interesting case that illustrates this interaction between thinking about things from an individual point of view and an individual evolutionary point of view, and also the ecological population level consequences of that trait and that evolutionary process. Okay. So when you think about this from an individual point of view, you can see there's clear differences straight away. So with a constitutive defense, you're never getting infected. That's great. With an induced defense, you're getting infected, and you've got a chance you might die while you're infected. So what I assumed is that there's different costs. So constitutive defense are great, but to have them, you've got to build them. So maybe it's a thicker gut wall, it's any of these things you're turning on. You have to have it all the time. So that means you're paying a cost for it even when there's no pathogen there. Okay. So that's the downside of this. You've got it, but you might not even be infected. But if you are challenged, you're not going to get infected. So from an individual point of view, that's great. You're not going to get infected. From an individual point of view, the bad thing is you're paying the cost for it, and there might not even be a pathogen around. Okay. This contrasts with induced defenses where, okay, you get infected, you might die before you manage to clear this thing. But the good thing is you're not paying the cost until you turn it on. Okay. So there's a difference from an individual point of view. But there's also a difference from what that means to the epidemiology of the disease. Right? So in this case, you're stopping infection. So infection and prevalence is going down. In this case, you're still allowing yourself to be infected, and therefore you could infect other individuals. So there's two differences. There's differences both at the bottom of at the level of the individual, and there's also differences in how it feeds back into the epidemiology. Okay. So we need to build an ecological and evo-ecological model to understand what's going to happen. So one of the things these models will tell you that the outcome, the relative investment in constitutive versus inducible defenses, will depend on resources in the environment, as those resources will then determine how dense and how large your population is. Okay. So there's a whole series of things and a whole series of predictions from these models, but I want to concentrate on this one. So at high resources, you've got high density, therefore there's a high risk of challenge, therefore it's worth paying the cost of constitutive defense all the time because there's a high chance you're going to get infected. Okay. So that tells you that you can't just look at the relative cost benefits to an individual. You have to understand it in the context of the ecology and the feedbacks on the ecology. Okay. So ecology matters to evolutionary outcomes. It's nice to test the predictions of these theories, so then you need a nice model system. And being at Berkeley, I'm contractually obliged to talk about CRISPRCas. So, but CRISPR-Cas was not invented at Berkeley. CRISPR-Cas was invented by bacteria. And CRISPR-Cas for bacteria is an immune system. And it's an immune system that I think is a really nice model for induced defenses. And the way in which I'm going to break down bacteriophage interactions, again, this is a conceptual model, is that you have defense to them can be surface modification, and this is a constitutive defense. It's going to stop you ever getting infected, and it's very costly in the absence of disease. Then CRISPR-Cas comes in, you whack you, and you defend yourself after being infected. Okay. And the nice thing, if you buy that as a good model system for this, it's bacteriophage. You can do experimental evolution. It's great. You can stick things in the fridge, come back, and they'll still be there. You can go back in time. If it all messes up, you can do it again. If PG&E shut the power to your lab for 5 days, you can just do the experiment again, and like in the insects and most things. So that's what we can do. So we could have high resources. We have low resources. We do an evolutionary experiment, and we can get a super clear response that absolutely fits our model. So we have two models now. So what we found is that at high resources, they invest in surface modification. At low resources, they invest in CRISPR. Okay. Which is great and totally fits the theoretical model. And although, you know, our model system is still just a model, it's nice to have an analog biological model test of the theoretical model. And again, straight away, what that's telling you is that ecology, in the sense of the environment and in resources, really matters in determining the evolutionary outcome. Resources really matter to the cost of resistance. Resources really matter to the density of the population. The density of the population really matters to the selection for the particular type of resistance. And we get into some kind of very theoretical theory for a minute, because I like to, but I'll be quick. So one of the things that's a little bit less intuitive than those results, I think, is the complex interaction you can have between ecology and evolution. So many people, when they taught evolution, think about things as fixed fitness landscapes. You're kind of taught this idea that things are evolving up to the top of these fitness peaks. And that's kind of the way that evolution works. Okay. But I think the thing about feedbacks and evolution and ecology is that these fitnesses will change depending on these ecological dynamics through time. So slowly this peak might change, so you end up at some sort of optimum because these peaks are dependent on these ecological feedbacks. And this is potentially important for something that's called evolutionary branching, where you can get situations where as these ecological feedbacks change, the peaks change, and you end up somewhere that's a fitness minimum, and then you branch, you go these two ways. So you generate diversity. And one of the things that I've been extremely interested in is how eco-evolutionary feedbacks lead to the generation of diversity in host parasite systems. And if you like things kind of just intuitively, I think resistance is an absolutely fantastic example of this. So when people are talking about why do eco-evolutionary feedbacks matter, resistance is one that's really clear to understand. You think about resistance, so you get an allele to increase your resistance in a population. It's going to spread in that population if the benefits of defending you from the disease outweigh the costs of having the resistance. But the key thing is, as it starts spreading and more individuals have this resistant allele, the prevalence of the disease in the population falls because more individuals are resistant. And therefore, the advantage falls as it spreads. Okay? And that's the ecological feedback, because it's the prevalence feeding back on the ecology, on the evolution. Therefore, selection for resistance falls, and we have direct negative frequency dependence due to the ecological, eco-evolutionary feedback. And that can lead to the generation of diversity. And I've been very interested in trying to predict what sorts of patterns and interactions will lead to the generation of diversity in host-parasite coevolution. And there's all sorts of possible outcomes that lead to lots of diversity, whether it's diversity through time, this is where the alleles are just varying through time, whether you end up at an optimum, or whether you branch and you end up with lots and lots of diversity. It's critically dependent on the ecology, and critically dependent on resources, which means if you have a whole load of different populations of a particular host-parasite interaction, and they've all got slightly different ecologies, or even predictable ecologies because they're on different latitudes and climes, they can all be at different parts of this co-evolutionary outcome. And if you imagine these populations are connected with gene flow, you can start to understand why there's so much diversity in ecology, in host-parasite interactions, just due to basic eco-evolutionary principles and diversity. Okay. All right. So that's the sorts of things we're interested in, all the way through to very basic theoretical questions. But we do try and apply some of our theory, and I'm just going to finish by, if Sylvie's computer can handle it, because my computer couldn't handle it earlier, the example that I came here to talk to Sylvie and Prin about. And that's in vector control. And lots of you understand what we do with vector control. We have vectors, we have individuals within inside houses, and this is talking about malaria, we're talking about night biting mosquitoes coming into the house and biting you. So what we try and do is protect people within the house by having bed nets and insecticide-treated bed nets and indoor residual spraying. So a lot of the protection is due to insecticide use inside your house. So when the malaria vectors are coming in to bite you at night, we're killing them. That's great. There you go. Dying. It worked. There's more of those to come. These are Penny's slides, not my slides. I should credit that. Okay. You know about DDT. DDT had lots of problems, so we banned DDT, mainly, I guess, for its use in agriculture. Do you agree with that? Actually, in vector control, DDT was incredibly effective. And one of the things about DDT is, as well as being an insecticide, it's an excellent spatial repellent. So it keeps the mosquitoes out of the house. Right? And since we've lost DDT, there's been a lot of interest in, can we develop spatial repellents? So how about if we just put chemicals on houses that keep the mosquitoes out? Okay, and this is a meeting on that. Lots of work going on. Lots of investment in Gates in trying to develop spatial repellents, because the problem is we don't have any really effective spatial repellents, things we can put on houses that will keep the mosquitoes out if we can't use DDT. So the approach is to get chemical companies to develop new products or new ways of maintaining them in the environment to keep spatial repellents. The thing that Penny asked was, well, we know there's going to be evolution. We know we're going to get insecticide resistance. Can we, in this case, use evolution to help us? Can we get evolution on our side? And we built this model which looked at what would happen if you combine this poor spatial repellent. We have a lot of those. We have a lot of really bad spatial repellents. That's easy. And an insecticide. Okay. With the argument that this could actually help us to develop, get evolution to help us to develop better spatial repellents and lead to better disease control. Okay, so these are Penny's great slides. So we have a model. It's a PopGen model. Anyone who recognises it, it'll be those sorts of equations. It looks a bit like that. Okay, we can make a bunch of assumptions. The model itself has a whole bunch of assumptions in it, some of which are very good because we know quite a lot about insecticide resistance, some of which are very, very bad because we don't know very much about spatial repellents and deflection. Okay, so we have a bunch of different cases. One is an untreated property where the mosquitoes just come in and they bite people who are very unhappy. Then we have an insecticide where the mosquitoes come in and that's great and we kill off the mosquitoes and that's brilliant, but you're causing a very large selective pressure for insecticide resistance. Okay. You could just have this repellent only, but as I said to you, they're really bad. So most of them are still coming in and still biting people. So they're happy, they're unhappy. Okay. And then this is what we propose, that we combine these two together. And what will be the outcome? So what's the argument? Well, intuitively, this is very easy. So what's happening is that you have both the spatial repellent and the insecticide. Initially, it's a rubbish spatial repellent, so loads of them come in, but then they all get killed by the insecticide. So what you've done is select for mosquitoes in the natural population which are repelled. Okay. So this is great. So you keep going on, do it again. A lower proportion are coming in, assuming you can get there's genetic variation for repellent and deflection in the population. Slowly they keep coming in, more and more are being repelled. And the other thing to notice is less and less are being killed by the insecticides. So the selection for insecticide resistance is also going down. Okay. So this is the idea. It's a fairly intuitive idea. But the thing that's cool about it as a potential new approach is that you don't have to change your current approach. You're already putting the insecticide in the houses. All we're saying is let's take this rubbish spatial repellent that we have, let's put that there as well. And then through time, can we select for deflection in the population? Then less and less individuals are going into the house. And we're also potentially protecting the insecticide for longer as well, till you get to this perfect situation. So what did we do? Well, we haven't done any data or anything yet, and that's what we're hoping to do. We're hoping to convince somebody to actually get data for us. But we can build models, and actually we know quite a lot about insecticide resistance. We don't know anything about the genetic basis of deflection, so we can use the nice thing about the models, though, is you can just run lots and lots of scenarios and say, do you think this is going to work? And the insights from our models are that this is going to work a lot of the time, and if nothing else, what it will do is lengthen the amount of time that you can keep one insecticide for, so you can change them much, much slower. But the key to actually getting someone to do this, I think, is to understand the evolution of deflection in wild populations, and hopefully that's what we're going to do. Okay, so the basic conclusion of that, if you combine the repellent and the insecticide, we can get the evolution of a better repellent. They are going to evolve, I've got no doubt about that. It's whether they do it quickly enough. Decrease selection for the insecticide resistance. And the nice thing is that you can add this to existing control programs. There's no point just coming up with a whole new way of doing something that's evolutionarily quite a sensible way of doing it if you have to utterly change the way in which you're currently controlling populations. All right, just finally to sum up, we've been very interested in how diversity is driven by coevolution. Very interested in optimal immune defense. I think there's all sorts of very interesting evolutionary questions about the evolution of tolerance and the impact on virulence of tolerance and the impact of immunopathology. A lot of the time, you're not killed or really damaged, particularly by the pathogen. You're damaged by your own immune responses. And understanding the evolutionary drivers of those is really interesting. I think we can use modeling to improve infectious disease management, and particularly if we start doing some sort of evolutionarily rational modeling or disease management. Take into account evolution is going to happen, how do we manage it, and when can we use it? And I think there's lots of opportunities for new intervention strategies from that. And I think these kind of eco-evo impacts of interventions are extremely important. So what happens at the population level as well as what happens at the individual level is really critical to the outcome. Okay, thank you.
All right, thanks very much, Mike, for that wonderful talk. Any questions? Well, I'd like to start out then. Just something that you were saying right at the beginning, where it's more easy to evolve resistance against any disease in a resource-rich environment.
Yeah.
Do you know if there's any data that we can see that in human populations now, where we have people living in more resource-abundant environments and people in less resource-abundant environments?
So I don't know about an evolutionary
Right.
impact of that. I don't know about that. I think, you know, this question of if you're in a better condition, you'll be better at fighting disease is pretty well established. But I don't know from an evolutionary point of view. One of the things I think is very interesting, though, is understanding the genetic variation in resistance or immunology, immune response in people is really interesting and understudied. So I remember we had a computational biology hire at Berkeley recently, and people were looking at that now with all these new genomes that are coming out to get at this problem about variation in defense in humans, variation in disease susceptibility. And they always start their talks with this introduction that says we all know that there's variation in terms of resistance in humans, and they say sickle cell, which is like 1950, right? And but I do think that with the number of new genomes that are coming in and the developments in this, we can potentially get at that and try and understand that it's clear there's huge amount of variation in humans in terms of resistance. But I don't think we've really had the data until now to understand that properly with models. But I do think it's underplayed. I do think there's vast differences between people in terms of their susceptibility.
Thanks, Sylvie. Nice talk. Thank you. Can you say a little bit about how you're accounting for variation in viral strains early on in terms of the modeling that you're doing and how you're thinking about capturing that part of the story?
Yeah, that's a good question. So if you look at most of the evolutionary theory in hosts and parasites, they just model the evolution of the parasite. And so I did a bunch of stuff just to model the evolution of the host. But actually the coevolution is super important. And the thing that's critically important to that is our understanding of specificity and the nature of specificity between genotypes of the host and the parasite. And we've done a bunch of very basic coevolutionary models, and that is absolutely critical to the predictions of when and how diversity evolves. So how specific those interactions are is absolutely critical. So I think that the challenge that we have is that it's really hard to get phenotypic data on host-parasite interactions because you've got to infect things and see if they die and how quickly they die. And then if you're trying to do that on the basis of genotypes, when there's specificity and variation in specificity, we really lack data. So we have data in plant systems, we have data in bacteriophage systems, but we lack that sort of data. And we have models, we have matching allele models or gene for gene models in plants, typically matching allele models in animals, but they capture that specificity in very simplistic ways. And how you model that specificity, we can show, is incredibly important to the outcome. But we don't have that much data outside of bacteriophage and plant pathogens. So it's a challenge. I don't think we understand the specificity, what determines specificity in mammals particularly.
All right, well, let's thank Mike again